Good evening. So we'll start today's class. I'll share my screen. Okay. Okay. So I have shared my screen. I hope I'm audible and my screen is visible. If I'm not, you are please requested to unmute and let me know. So good evening and welcome back to the 11th live session of the series of live sessions that we have for this course RNA Biology. So what we do in these live sessions, as we all know, that we discuss some pertinent, relevant questions regarding the course, and we not only discuss the questions, but we also try to discuss answers to those questions and similar type of the entire concept behind it, basically, so that it gets easier for us to solve similar type of other questions, right? And we try to keep the sessions as interactive as possible. So if you have any doubt or if you know the answer, please you can unmute and let me know, or maybe raise a hand, I write in the chat box, anything is fine with me. Okay, so we'll start with the first question for today. Proteins are able to act as better enzymes than RNA. Is it true or false? I'll wait for a few minutes so that you can think about the answer, then we'll discuss the question. There's no issue, right, with the video call? Okay, I hope not, yeah. So if anybody knows the answer, you can let me know, or we'll discuss after a few minutes. Okay, so the answer will be true. At first, it was believed that proteins can be only enzymes, but then RNA can also act as biological catalysts. And so the gist is basically that while RNA act as enzymes like ribozymes, proteins are generally considered better enzymes due to their greater diversity of chemical functionalities and the ability to form complex 3D structures that facilitate catalysis, right? So, proteins with their 20 different amino acid side chains offer a wider range of chemical functionalities compared to RNA, which is limited to 4 nucleotides. And this allows proteins to engage in a broader spectrum of chemical reactions and catalysis. Proteins can fold into complex 3D structures, creating active sites that perfectly bind to substrates and facilitate catalysis. RNA structure is more limited, making it less versatile in this regard. While ribozymes can catalyze reactions, protein enzymes generally exhibit higher catalytic rates and efficiencies. Proteins often utilize coenzymes like vitamins or metal ions to expand their catalytic capabilities, further enhancing their versatility. This transition from an RNA world to a protein dominated world likely occurred because proteins offered greater catalytic capabilities and adaptability, right? So, the example, if you want, ribosome, a complex RNA protein structure, plays a crucial role in protein synthesis, demonstrating the catalytic capabilities of RNA. Whereas for protein enzymes, enzymes like pepsin, amylase, and lipase are essential for breaking down food molecules, showcasing the power of protein enzymes in biological processes. Now, what are the limitations of RNA as an enzyme? RNA's chemical functionalities are limited, restricting its catalytic lifestyle potential. RNA structure is less versatile, making it difficult to form complex active sites. And ribozymes generally exhibit lower catalytic rates compared to protein enzymes. Right? So we've all talked about gene structures. We are not going into the very much detail of gene structures in eukaryotes or prokaryotes. But just know that this is how a DNA would look like. Okay, it has a regulatory sequence, a promoter, enhancer, silencer, and all of that. Open reading frame and 5 prime untranslated region, 5 prime UTR, 3 prime UTR, all of that. Once DNA gets replicated to DNA, once it gets transcribed to mRNA, there are the exon sequences and the intron sequences. Okay, and the 5 prime UTR and 3 prime UTR. Other things are not translated, transcribed. Then from pre-mRNA we go into the mature mRNA, which happens after splicing. So the introns, which are the non-coding regions, are spliced out and the exons are stuck together, and a 5 prime cap is added to the and a cap is added to the 5 prime end, and a poly A tail is added to the 3 prime end. That makes the mature RNA, mRNA. So you've talked about transcription in eukaryotic cells previously, but if you see, this is how translation is all about. Translation happens in cytoplasm. Replication and transcription will happen inside the nucleus. If you see over here, then mRNA gets transported out of the nucleus via the nuclear envelope into the cytoplasm, where there is ribosome and amino acid tRNA and all of that, which will help in the process of translation, that is conversion of mRNA to protein. Okay, so transcription we have discussed in detail in some of the previous classes, and also it's a very complex process. There are a lot of elongation factors, initiation factors, and everything required. To get the best knowledge of all these processes, I request you to go through books like Watson and all of that, molecular biology books. It will give you a very detailed overview of these processes. Okay, then we have transcription factors, promoter, transcription starts, guide, gene, RNA polymerase, etc. Okay, this is how transcription, direction of transcription and everything. We have talked about translation, how the translation happens in cytoplasm after the mRNA is transported out. Okay, so amino acids corresponding to the codons are added to the growing protein chain, right? And the mRNA transcript code is added, and the protein is constructed, code residing on mRNA translation constructing protein. Now, tRNA released after amino acid removed growing polypeptide chain, tRNA from the cytosol carry amino acids. The translation process incorporates 12 different amino acids in the precise sequence detected by the 3 base codons built from an alphabet of 4 bases. The process in the ribosome builds the polypeptide chains that will become proteins. This is translation. We have talked about the central dogma of molecular biology, which is an explanation of the flow of genetic information within the biological system. So, the central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information, which states that such information cannot be transferred from protein to either protein or nucleic acid. So, DNA is replicated on itself, so DNA, then DNA to RNA, RNA to protein. So Jim Watson is the first edition of the molecular biology gene describes the simple version, okay? So what is a gene? First mention of gene was made by Mendel. He explained his results in terms of discrete inherited units, now known as genes, that give rise to observable physical characteristics, physical genotype and phenotype. And in 1905, Johansen introduced the term gene, and in 1908, Bateson term genetics. Today you have learned that a gene, a DNA segment, contains information for a protein that mediates gene function, right? And this is central dogma of molecular biology. DNA makes RNA and RNA makes protein. Now we know much more than 1950s to 1960s, right? Is this central dogma still relevant? Not really. There are some exceptions that are listed below. Some gene may have splice variants with distinct regulatory functions like tobacco N gene encoding TMP resistance. Multiple subunits of an enzyme could be encoded by a number of genes. tRNA genes for tRNAs involved in protein synthesis. sRNA genes for sRNAs involved in splicing PMRNAs. Ribosomal genes for rRNAs involved in protein synthesis. MicroRNAs with regulatory functions. Prions can make more prions from normal proteins. Right? So central dogma is not 100% relevant today, but then 99% still relevant. So if metabolites are closest to phenotypes, so genes to mRNAs, mRNAs to proteins, proteins to metabolites, and then phenotypes, right? So proteins are composed of 20 amino acids. Different proteins are made by combining these 20 amino acids in different combinations, and they are manufactured by the ribosomes. What are the functions of proteins? Helps fight disease, help build new body tissue. Enzymes used for digestion and other chemical reactions of proteins. Enzymes speed up the rate of reaction. Component of all cell membranes. So if you talk about making proteins, step 1 is transcription. Copying of genetic information from DNA to RNA called transcription. Why? Because DNA has genetic code for the protein that needs to be made, but proteins are made by the ribosomes. Ribosomes are outside the nucleus in cytoplasm. DNA is too large to leave the nucleus, double-stranded. RNA can leave the nucleus, it is single-stranded. Now, part of DNA temporarily unzips and is used as a template to assemble complementary nucleotides into messenger mRNA. mRNA then goes through the pores of the nucleus with the DNA code and attaches to the ribosome. If we talk about making proteins, the translation, decoding of mRNA into a protein is called translation. Transfer RNA carries amino acids from the cytoplasm to the ribosome, and these amino acids come from the food we eat. Proteins we eat are broken down into individual amino acids and then simply rearranged into new proteins according to the needs and directions of our DNA. A series of 3 adjacent bases in an mRNA molecule codes for a specific amino acid called a codon. Each tRNA has 3 nucleotides that are complementary to the codon in mRNA. and each tRNA codes for a different amino acid. So RNA carrying the DNA instructions and tRNA carrying amino acids meet in the ribosome, right? So the messenger RNA is transcribed in the nucleus, and the mRNA then enters cytoplasm and attaches to the ribosome, right? Over here, if you can see, it is coming out at ribosome. Translation begins at AUG, start codon. Each transfer RNA has an anticodon whose bases are complementary to codon on the mRNA strand. The ribosome positions the start codon to attract its anticodon, which is part of the tRNA that binds methionine. The ribosome also binds the next codon and its anticodon. Amino acids are joined together to make a protein. The polypeptide assembly line. The ribosome joins the two amino acids methionine and phenylalanine and breaks the bond between methionine and its tRNA. The tRNA floats away from the ribosome, allowing the ribosome to bind another tRNA, and the ribosome moves along the mRNA, binding new tRNA molecules and amino acids. And then the process continues until the ribosome reaches one of the three stop codons, and the result is a complete polypeptide. This is genetic code table. I think I have shown it to you in many of the previous classes. EUGs, methionine, AACs, asparagine, GACs, aspartic acid, and UAA stop codon. Central dogma. Now, DNA we know, blueprint of life. Structure we know it's polymer of nucleotides composed of ATGC, 4 DNA bases, adenine, purine bases, and pyrimidine bases. Complementary base pairing is adenine pairs with thymine and guanine pairs with cytosine. There are 2 double bonds between adenine and thymine and 3 double bonds between cytosine and guanine. You know about the DNA structure, it's a double helix, Watson and Crick. DNA replication in the nucleus. Each DNA strand becomes separated and parent strand becomes apart and proper base pairs are separated on the template. Okay, so we not going to very much detail about these question answer sessions. You know about the central dogma. You know about this. You know about genetic code. 20 amino acids, building blocks of protein. Okay. So what are the steps in translation? mRNA leaves the nucleus and migrates to ribosome. mRNA binds to small ribosomal subunit. tRNA brings the amino acid to the ribosome, where anticodon on the tRNA binds to the codon of the mRNA. The amino acid bonds to its adjoining amino acid to form a growing polypeptide molecule. The tRNA without the amino acid is released from the ribosome. Other tRNAs bring amino acids into the ribosome to complete the protein molecule. Protein translation, protein synthesis. Amino acids are the repeating subunits of protein molecules. The order determines the protein. Okay. So summary, biological activity of protein depends largely on its 3D structure, which also depends largely on the primary level structure, primary level sequence. So this is the protein summary. Okay. So in cell nucleus, RNA is produced by transcription. RNA single stranded substitutes the sugar ribose for deoxyribose and the base uracil for thymine. Messenger RNA or mRNA conveys the DNA recipe for producing cell cytoplasm. mRNA binds to ribosome. Each 3 base codon of the mRNA links to a specific form of transfer RNA containing the complementary 3 base sequence. This tRNA in turn transfers a single amino acid to a growing polypeptide protein chain. Each codon directs the addition of one amino acid to the protein. Same amino acid can be added by different codons. In this illustration, the RNA sequences are both specifying the addition of the amino acid alanine. Both DNA and RNA have a direction. One end is the 3', other end is the 5'. Thus, codons are read in one direction only. Also note there is a redundancy in genetic code. That different sequences can specify the same amino acid, right? If there are mutations that may lead to changes in the sequence, they may pass around to future generations. So point mutation is a single base substitution, deletion a small DNA segment is lost and insertion a segment of DNA is added. Or there can be frame shift mutations, modification of the reading frame after deletion or insertion, resulting in all codons downstream being different. Now, if we talk about enzymes are nothing but biological catalysts produced by the living cells, and they catalyze several reactions in the body. They're protein by nature. That's a wrong statement because they can be RNA as well. They're specific in its action. Each enzyme can catalyze only one type of reaction. They are required in very small quantities, and the loss of catalytic activity was observed when they are subjected to heat or strong acid or organic solvents. Now, the enzymes mainly catalyze the metabolic pathways in the body. The deficiency of the enzyme leads to inborn errors of metabolism. Most of the enzymes are produced by the cells of a particular tissue and function within that cell. Such enzymes are called intracellular enzymes. On the other hand, there are certain enzymes which are produced with cells of a particular tissue from where these are liberated for use in other tissues. Such enzymes are called extracellular enzymes. The enzyme binds with its specific substrate and forms an enzyme-substrate complex. At the end of the reaction, the substrate is converted into the product and the enzymes remain unchanged. In enzymatic reactions, the molecules at the beginning of the process called substrates are converted into different molecules called products. Almost all chemical reactions in biological cell needs enzymes. Now, like all catalysts, enzymes work by lowering the activation energy for a reaction. This increasing the rate of the reaction. As a result, products are found faster and reactions reach the equilibrium state more rapidly. So most enzyme reaction rates are millions of times faster than those of comparable uncatalyzed reactions. Enzymes are not consumed by the reactions they catalyze, nor do they alter the equilibrium of these reactions. Enzymes are known to catalyze about 4,000 biochemical reactions. A few RNA molecules called ribozymes also catalyze reactions, with an important example being some parts of the ribosome. Enzyme activity can be affected by other molecules. Inhibitors are molecules that decrease enzyme activity. Activators are molecules that increase activity. Many drugs and poisons are enzyme inhibitors. Activity is also affected by temperature, chemical environment, and the concentration of the substrate. Now, some enzymes are used commercially, for example, in the synthesis of antibiotics. Some household products use enzymes to speed up biochemical reactions. So, enzymes in biological washing powders break down protein or fat stains on clothes. Enzymes in meat tenderizers break down protein in smaller molecules, making the meat easier to chew. Now, the enzyme remains confined to a particular area of cell body, which makes it exclusive. For instance, fatty acid synthesis takes place in cytosol, whereas fatty acid oxidation takes place in mitochondria. You can see these are different organelles and the different enzymes in the organelles. Okay. Now, if we talk about the history of enzymes, as early as the late 17th and as early as the late 17th century, as the digestion of meat by stomach secretions and the conversion of starch to sugars by plant extracts and saliva were known. However, the mechanism by which this occurred had not been identified. So, in the 19th century, when studying the fermentation of sugar, to alcohol by yeast, Louis Pasteur came to the conclusion that this fermentation was catalyzed by a vital force contained within the yeast cells called ferments, which were thought to function only within living organisms. And then 1897, Edward Buchner submitted his first paper on the ability of yeast extracts that lacked any living yeast cells to ferment sugar. So in a series of experiments at the University of Berlin, he found that the sugar was fermented even when there were no living yeast cells in the mixture. So he named the enzyme that brought about the fermentation of sugar sucrosimase. And in 1907, he received the Nobel Prize in Chemistry for his biochemical research and his discovery of cell-free fermentation. Following Buchner's example, enzymes are usually named according to the reaction they carry out. Typically, to generate the name of an enzyme, the suffix "ase" is added to the name of its substrate. Like lactase is the enzyme that will cleave lactose or the type of reaction. DNA polymerase forms DNA polymers. So James B. Sumner showed that the enzyme urease was a pure protein and crystallized it. Sumner did likewise for the enzyme catalase. Northrop and Stanley, who worked on the digestive enzymes pepsin, trypsin, and chymotrypsin, these two scientists were awarded the 1946 Nobel Prize in Chemistry. So most enzymes are protein in nature. Some enzymes require the presence of certain additional organic or inorganic substances and are conjugated proteins. Such enzymes are called holoenzymes. The protein part of the conjugated protein is called apoenzymes, and the non-protein part is called the prosthetic group. Several apoenzymes require the presence of metal ions such as magnesium 2 plus as hexokinase, there is zinc 2 plus with the activity of carboxypeptidase. Such inorganic ions are called cofactors. If the metal ion is the integral part of the enzyme, such enzymes are called metal enzymes. So in biology, the active site is a small port in an enzyme where substrate molecules bind and undergo a chemical reaction. The active site is usually found in a 3D groove or pocket of the enzyme lined with amino acid residues. So these residues are involved in recognition of the substrate. After an active site has been involved in a reaction, it can be used again. Enzymes can act in several ways, all of which lower the energy of activation. So lowering the activation energy by creating an environment in which the transition state is stabilized. Substrates need a lot of energy to reach a transition state, which then decays into products. The enzyme stabilizes the transition state, reducing the energy needed to form the products. Now, if we talk about the lock and key model, enzymes are very specific and it was suggested by the Nobel laureate organic chemist Emil Fischer in 1894. This was because both the enzyme and the substrate possess specific complementary geometric shapes that fit exactly into one another. This is often referred to as the lock and key model. However, while this model explains enzyme specificity, it fails to explain the stabilization of the transition state that enzymes achieve. The favored model for the enzyme-substrate interaction is the induced fit model. This model proposes that the initial interaction between enzyme and substrate is relatively weak, but that these weak interactions rapidly induce conformational changes in the enzyme that strengthen binding. Substrate entering the active site of enzyme-substrate complex, enzyme changes shape slightly as substrate binds. Enzyme product complex, products leaving active site of enzyme. Now what about the Michaelis constant and Michaelis-Menten constant? As enzyme catalyzed reactions are unsaturated, the rate of catalysis does not show a linear response to increased substrate. The initial rate of the reaction is measured over a range of substrate concentrations denoted as S. The reaction rate V increases as S increases. However, as S gets higher, the enzyme becomes saturated with substrate and the rate reaches Vmax, the enzyme's maximum rate. The Michaelis constant Km is experimentally defined as the concentration at which the rate of the reaction is half Vmax. Okay, this is Michaelis maintain this is V naught. This is where the Vmax is substrate concentration on S. Now, enzymes can be classified into 6 groups according to the International Union of Biochemistry (IUB). Can be oxidoreductases that are involved in oxygen reduction reactions like lactase, lactate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, or transferases that transfer a particular group from one substrate to another, alanine aminotransferase, or hydrolases. Hydrolases substrate with the addition of water molecule like glucose 6-phosphatase, amylase, pepsin, or lyase catalyze the removal of a small molecule from a large substrate without the addition of water. Fumarase or enolase, isomerase, the isomerized substrates, or ligase synthesize substrates by joining two substrates with the utilization of energy. Now the EC number, enzyme function number, is a numerical classification scheme for enzymes based on the chemical reactions they catalyze. Every EC number is associated with a recommended name for the respective enzyme. Every enzyme code consists of the letters EC followed by 4 numbers separated by periods. Okay, we are not going into too much detail of all of this. And the stereotoxicity is the group of enzyme catalyzes either L or D isomer. Reaction specificity will be one enzyme catalyzes only one type of reaction. And substance specificity is absolute specificity, like glucokinase acts on glucose only, or hex group specificity, hexokinase catalyzes hexoses. If we talk about bond specificity, refers to the action of the proteolytic enzymes which act on peptide bonds on proteins. For dual specificity, there are 2 types of dual specificity. The enzyme act on 2 substrates by 1 reaction types. Xanthine oxidase enzyme acts on xanthine and hyperxanthine, 2 substrates by oxidation 1 reaction type, and the enzyme act on 1 substrate by 2 different reaction types. Isocitrate dehydrogenase enzyme acts on isocitrate by oxidation followed by decarboxylation, 2 different reaction types to produce alpha ketoglutarate as a reaction product. Now, what are the factors affecting enzyme activity? If you talk about the effect of pH, each enzyme has an optimum pH at which the activity of the enzyme is maximum. Either decrease or increase pH causes a decrease in enzyme activity. Examples of optimum pH are these. Okay, pepsin has an optimum pH of around optimum pH of amylase 6.8, ALP alkaline phosphatase is 9. Okay, all of these. Now, effect of temperature, the temperature at which the enzyme activity is more is called the optimum temperature. Enzyme activity was duplicated for every increase in temperature by 10 degrees, still reaching the maximum velocity for the enzyme activity at the optimum temperature. So after this point, it was seen that the enzyme activity decreases with the increment of temperature. So 100 degrees Celsius, the enzyme will be denatured. While at 0 degrees, the enzyme will be kept inactivated, and when the temperature increases, the activity of the enzyme will be increased. Optimum temperature of enzymes in the human body is mostly 37 degrees Celsius because that is the physiological temperature. Effect of substrate concentration. So at low substrate concentration, enzyme molecules are free initially and the ES complex formation is proportional to the substrate concentration. At higher concentration, all the enzyme molecules are saturated with substrate. So there will be no change in the activity further. Hence, in an enzyme reaction subsystem, more substrate is taken than the requirement. The velocity of the enzyme reaction is directly proportional to the enzyme concentration. There are various enzyme inhibitors. I will not go into the details of these inhibitors. If you have time at the end of the class, we will go into it. So coming back, these are all regarding enzymes. We'll come to it if we have time at the end of the class, but it's too much detail for the question that we started with. So the question that we started with was proteins are better sorry, proteins are better enzymes. If we talk about ribozyme, which is an RNA enzyme, okay. So before the discovery of ribozymes, the enzymes which are defined as catalytic proteins were only known to be biological entities. So first ribozymes were discovered in 1980s by Thomas Sheck, who was studying RNA splicing in the ciliary protozoan Tetrahymena haemophila, and Sidney Ortman, who was working on the bacterial RNA-sp complex. So the ribozymes were found in an intron of an RNA transcript which removed itself from the transcript, as well as in the RNA component of the RNA-sp complex, which is involved in the maturation of pre-tRNAs. So ribozymes often have divalent metal ions such as Mg2 plus cofactors. So ribozyme is an RNA molecule that catalyzes a chemical reaction. And many natural ribozymes catalyze either hydrolysis of one of their own phosphodiester bonds or hydrolysis of bonds in other RNAs, right? So they have also been found to catalyze aminotransferase activity of the ribozyme. So investigators studying the origin of life have produced ribozymes in the laboratory. They are capable of catalyzing their own synthesis under very specific conditions, such as RNA polymerase ribozymes. So ribozymes may play an important role as therapeutic agents, as enzymes which tailor defined RNA sequences as biosensors, and for applications in functional genomics and gene discovery. So although most ribozymes are quite rare in the cell, their roles are sometimes essential to life. For example, functional part of the ribosome, the molecular machine that translates RNA into proteins, is fundamentally a ribosome, composed of RNA tertiary structural motifs that are often coordinated to metal ions such as Mg2 plus cofactors. So there is no requirement for divalent cations in a 5 nucleotide RNA that catalyze transphenylation of a 4 nucleotide substrate, which has 3 base complementary sequence with the catalyst. So basically, enzymes are proteins that act as biological catalysts to speed up rate of chemical reactions without being consumed in the process. The active site of the enzyme is where the substrates bind, and substrates are the reactants in a chemical reaction requiring enzymes. Okay. So if we talk about the lock and key model, in the older model, the enzyme binding was an exact match for the substrates. However, in the more modern induced fit model, the enzyme binds most tightly to an intermediate form of substrate or product, right? And this model is more compatible with reversible reactions that are catalyzed by one enzyme. We have talked about naming of enzyme specificity, we have talked enzyme activation, about coenzymes, we have talked about all the factors. I'll not go into much detail now only. Okay, so coming back to the question that we started with, proteins are better able to act as better enzymes in RNA, which is true. Okay, so if you have any doubt regarding this question, you can ask me now or write in the chat box. I'll wait for a few minutes and we'll move on to the next question for today. Okay, so we'll move on to the next question for today. Which is not a ribozyme? RNSP, hammerhead, HDV, or HIV? Which one do you think is not a ribozyme? I'll wait for a few minutes for you to answer the question, then we discuss the answer. Okay, so the answer will be HIV. Okay, so if we talk about ribozymes, I have already talked RNA persist catalytic activity. It increases the rate and specificity of phosphodiesterase bond cleavage, peptide bond synthesis, widespread occurrence in nature from viruses to humans. Okay, and 1989 Nobel Prize in Chemistry has been awarded to Sidney Altman and Thomas Cech for the discovery that RNA in living cells is not only a molecule of heredity but also can function as a biocatalyst, right? So this is a natural occurring ribozymes. I've already discussed for RNA, tRNA, peptide bond formation, protein synthesis, ribonuclease, P-phosphorescent bond hydrolysis, maturation or transfer, RNA cell splicing, and all of that. Obviously, there is a hairpin ribozyme, which is a phosphorescent bond hydrolysis. Now, structural features affect how RNA can function. RNA contains only 4 nucleotide bases compared to 20 amino acids found in proteins. Okay, it's a small repertoire of functional groups in RNA. High density of negative charges, localization of bases in the interior duplexes, X amino acid side chains are direct output from the polypeptide backbone, right? So, nevertheless, the mechanisms of catalysis are diverse and explored. Metal ions, acid base mechanism, small metabolite as a cofactor, substrate assistance. Usually, ribozyme combines several of these strategies. The catalytic strategies appear to be similar. RNA as well as protein enzymes use acid-base groups and metal ions to activate nucleophiles and to stabilize developing charge in the living group. And a ribozyme also requires formation of a specific secondary and tertiary structure of RNA by base pairing of complementary regions. Specific primary structure of certain regions is also necessary. Now, some ribozymes can speed up the rate of reaction 10 to the power 3, 10 to the power 11 times. HDV ribozyme cleaves the phosphodiesterase bond as fast as RNase. RNase-P catalyzes site-specific hydrolysis of precursor tRNA, which is essential for the formation of mature tRNA. Catalytic activity depends on the presence of divalent cations. Large ribozyme composed of both RNA and proteins, however, RNA moiety alone is the catalyst. For example, spread cell splicing introns, large introns that are able to splice out the cells in bacteria as well as eukaryotes. Splicing introns segments of non-coding RNA that are interspersed among the regions of mRNA that code for protein. Prior to translation, introns must be removed from a mature mRNA. Now, if we call cell splicing, unlike common introns, cell splicing introns can splice themselves out of pre-mRNA without the need for the spliceosome. Okay, although cell splicing introns can remove themselves from RNA in the absence of any protein in vitro, in many cases in vivo, cell splicing proceeds in the presence of certain proteins that increase the efficiency of splicing. Cell splicing introns mediate only one round of RNA processing, unlike protein enzymes. Now, group 1 introns cell splicing is initiated by the nucleophilic attack of 3' OH of an exogenous guanosine on the phosphodiesterase bond. Group 2 introns nucleophilic attack is realized by 2' OH of a specific adenosine within the intron. Metal lines are proposed to promote the formation of the correct active site structure, correctly position the substrate, activate the nucleophile by deprotonating the 2' OH of guanosine, and stabilize the negative charge. A group 1 introns 3' OH of an exogenous G attacks the phosphodiesterase bond at the 5' splice site. This bond is being cleft. G fuses to the 5' end of intron for transesterification. The free 3' end of the exon attacks the bond at the 3' splice site. This fuses the two exons and releases the intron. That is second transesterification. Group 1 introns denucleated binding site and group 2 introns at internal adenosine. G attacks the phosphodiesterase bond in the 5' splice site. Cleavage between 3' end of exon and 5' end of intron. Internal A attacks the phosphodiesterase bond at the 5' splice site. A terminal 3' merge of exon 1 attacks and leaves a phosphorylation bond at the 3' splice site. A new bond is formed between the two exons. Intron is released. Now, what is the importance of being folded? Specific primary, secondary, and tertiary structure is necessary for recognition of the guanosine binding site, recognition of the sites of splicing. This is the RNA hairpin. Now, group 1 introns are real enzymes. Cell splicing introns mediate only one round of RNA processing, but only once a group 1 intron has been spliced out, it can act as a real enzyme. It can rapidly recognize the complementary sequence of another RNA molecule, attack it by 3' OH of the bound G nucleotide, and catalyze its cleavage. Now we can change the IGs and thus generate tailor-made ribozymes that cleave RNA molecules of our choice candidate method for human therapy. Currently, synthetic ribozyme that destroys mRNA code encoding the receptor of vascular endothelial growth factor or VEGF is being readied for clinical trials. VEGF is a stimulant of angiogenesis and blocking its action may help starve cancers of their blood supply. Now, small ribozymes are viriated satellites like hammerhead, hairpin, and HTV, hepatitis delta virus ribozyme. Satellites, small RNA viruses or RNA molecules, their multiplication depends on the mechanisms of a host cell and the co-infection of a host cell with the help of virus. Ribozyme is a part of larger RNA that is being replicated by host RNA polymerases. The product of the replication is being self-cleft by ribozyme activity into unit-length RNA molecules. Nucleophilic attack of 2' OH on the neighboring 3' phosphate, forming 2' 3' cyclic phosphate. Probably an acid-base mechanism. 2' OH is activated for nucleophilic attack by abstraction of a proton by a basic group. Another proton is donated by an acid to stabilize the developing negative charge on the leaving group oxygen. Now in HTV, cytosine acts as an acid to protonate the leaving group and a divalent metal ion activates the nucleophile. Hammerhead ribozyme. Hammerhead and hairpin ribozymes can be found in several satellite RNAs associated with RNA plant viruses. HDV is a human pathogen. Co-infection of HDV with HPV is more severe than infection of HBV alone. Elements of bacterial mRNA that control gene expression via binding of small molecules. Okay. GLMS ribozyme located in the 5' UTR region of mRNA encoding glucosamine 6-phosphate synthetase. In the presence of GLCN6P, it releases its own mRNA. It downregulates the production of synthetase. Riboswitches may have function as metabolite sensors in primitive organisms. Now, conformational metabolite binding induces a conformational change in RNA that affects transcription, termination, translation, initiation. Now, GLCN6P amine might serve as an acid to activate the leaving group cleavage, right? So, ribosome is a ribozyme. Peptide transferase is a ribozyme. Whatever action is happening and the amino acid is being formed, the main catalytic activity is by an RNA, a ribozyme, right? If you see over here, this is how the amino acid tRNA brings the particular amino acid, matching the codon on the mRNA. Peptide synthesis is happening and translation is happening. So this is happening because of the RNA component. So the biological catalyst in ribosome is an RNA. So it's a ribozyme. So peptide transferase activity can be enhanced by protein L27. However, even in the acid-nothing proteins, reduced activity can still be observed. Although this protein facilitates peptide bond formation, it is not essential for peptidal transferase activity. Right? Give me a minute to wait for charging. Give me a minute. Okay, so although this facilitates propeptide bond formation, it is not essential for a peptidal transferase activity. Now, how does RNA catalyze peptide bond formation? Base pairing between the CC end of tRNAs in the P and A sites and 23S rRNA help to position the alpha amino group of amino acid tRNA to attack the carbonyl group of the growing polypeptide. So, proton transfer from the amino group of the amino acid tRNA via 2' OH of adenosine to its O3 prime transfer to amino acid tRNA, right? So RNA initially served both as a genetic material and a catalyst. Later, catalytic functions of many RNA molecules were taken over by proteins. So cationic clades such as monoamide can promote the polymerization of RNA-like monomers into RNA chains. RNA is the primary substance of life. DNA and proteins are later refinements. So cofactors used by ribozymes include vitamin B12, FMN, glucosamine 6-phosphate. Some of them are used by protein enzymes for oxidation, reduction and CC bond formation. So why do you have protein catalysts? Group 1 intron active site is mechanistically equivalent to DNA and RNA polymerases. So what selective pressure led to the current protein-based system for replication and transcription? The result might be greater fidelity, processivity, reaction rates, functional repertoire, etc. So which is not a ribozyme? HIV is the correct answer. Okay. If you have any doubt on this question, I'll wait here for 5 minutes. Then we'll move on to the next question for today. move on to the next question for today. Coupled transcription translation is for A. Increasing rate of protein synthesis, B. Facilitating transcription termination, C. Transcription initiation, and D. To create RNA secondary structure formation. I'll wait for a few minutes. You guys can tell me which one do you think is the correct answer. Okay, so the answer will be increasing the rate of protein synthesis, right? So the process of synthesis of proteins from messenger RNA after the transcription of DNA to RNA. So it takes place in cytoplasm by specialized organelle known as ribosomes. We have discussed all of these, right? Okay. So there are no endoplasmic reticulum in the prokaryotes, and ribosomes are suspended in cytoplasm, whereas endoplasmic reticulum are present in eukaryotes which harbors ribosomes. So translation takes place on rough endoplasmic reticulum in eukaryotes, whereas translation occurs freely in cytoplasm in the prokaryotes. So the codons of the mRNA are translated into amino acid sequence which leads to the synthesis of proteins. A translation requires a variety of cellular components such as proteins, RNAs, and different small molecules. It has three main steps: formation of RNA ribosome complex, formation of polypeptide chain complementary to the mRNA, and termination of a polypeptide chain. We have talked about the central dogma, so I'm not going to get into it. We have talked about different proteins, these are proteins. Okay, so George Weedle and Edward Tatum established a link between genes and enzymes in studying bread mold and Neurospora crassa. Okay, so from the experiment, they concluded that one gene produces one enzyme theory. Later, it was modified, obviously, because that's not more relevant today. One gene produces one protein, one gene produces one polypeptide chain. So DNA is transcribed to mRNA in the cytoplasm in prokaryotes as it has no nucleus, and in the nucleus in eukaryotes. So with mRNA and help of other RNA as well as protein molecules, mRNA is translated into specific protein cytoplasm. So therefore, RNA is the intermediate between DNA code and the actual synthesis of a protein. Okay, RNA protein synthesis. Now it carries code from DNA to ribosomes. It actually acts as a transporter of genetic information to protect this specialty. It is specific for a particular protein. Now, three types of RNA help in protein synthesis. Ribosomal RNA made in the nucleolus of eukaryotes, while it is made in cytoplasm in prokaryotes. It has two subunits. It combines proteins to form ribosomes. Prokaryotic ribosomes are of different size than eukaryotic ribosomes, 330S and 50S subunits in prokaryotes and 40S and 60S in eukaryotes. Some antibiotics target infected ribosomes in the host. Now, if we talk about the endosymbiotic theory, it states that some of the organelles in eukaryotic cells were once prokaryotic microbes. Three types of RNA help in protein synthesis. Transfer RNA anticodon sequence are present. Anticodon sequence matches codon on mRNA to add correct amino acid during protein synthesis. Amino acid alanine synthetase enzyme can digest a specific amino acid using energy from ATP. Now, what is the difference between protein synthesis in prokaryotes and eukaryotes? Prokaryotes do not have introns like eukaryotes. There is no or very little RNA processing in prokaryotes, and transcription and translation can be simultaneous in prokaryotes. So, protein synthesis requires genetic information as genetic code contained in the DNA. Now, genetic code, we know the amino acids, there are 64 codons, 61 code for amino acid, 3 for stop and 1 for start. The genetic code, this is all translation of codons into a polypeptide. So why genetic code triplet? Given that there are 4 bases in DNA and 3 is code for 20 amino acids, what is the basis for the genetic code? If one base, one amino acid, possible amino acids 4. If two bases, one amino acid, then possible amino acids 16. If three bases, one amino acid, possible amino acids 64, right? So the existence of a three base code was confirmed by experiments by Francis Crick and his colleagues. And the triplet code is degenerate, which means that each amino acid is coded by more than one triplet. So how are the codons matched to amino acid? Charging of tRNA by an enzyme called amino acid tRNA synthetase, also called tRNA ligase. One amino acid tRNA synthesis is present for each standard amino acid. 20 amino acid tRNA synthetases are present in most of the cell. Now what we call charging of tRNA? Charging of tRNA is accomplished by an enzyme amino acid tRNA synthetase. Enzyme which bonds amino acid to tRNA. The bond requires energy. ATP to AMP. Bond is unstable, so it can release amino acid at ribosome easily. So there is ester bond between amino acid and tRNA. So how are the codons matched to amino acid? So each codon represents a particular amino acid and each codon is recognized by specific RNA. tRNA. So only codon specific anticodon containing tRNA interacts with codon. Otherwise, tRNA of wrong anticodon cannot interact with codon site. So ribosomes are a complex of ribosomal RNA and proteins. It is composed of two subunits of unequal size, large subunit and small subunit. So it facilitates coupling of tRNA anticodon to mRNA. So it has many sites meant for specialized function, which are A side, which is the amino acid tRNA side. It holds tRNA carrying next amino acid to be added to the polypeptide chain. Then you have the P side, peptidal tRNA side. It holds tRNA carrying polypeptide chain. And then the E side, it ensures empty tRNA leaves ribosomes from exit side. Now what are the three steps? Initiation, elongation and termination. It brings together mRNA ribosomal units and initiated tRNA. Elongation in this phase, adding of amino acids takes place based on codon sequence. Termination here, codon ends and translation stops. Now what is the initiation of translation? In prokaryotes, protein synthesis begins with the formation of an initiator complex, which involves a small 38 ribosome, the mRNA template, three initiation factors that help ribosome assembly correctly, guanosine triphosphate GTP that acts as an energy source, and a special initiated tRNA carrying the end formyl methionine or FMET tRNA MET. Okay, for the Scott. Okay. So elongation of polypeptide chain. So numerous ribosomes translate same mRNA at the same time. That is the polyribosomes or polysomes. 3D folding chaperones provide essential kinetic assistance to protein folding by binding non-native proteins. Now, elongation of polypeptide chain. Elongation starts with the occupation of methionine tRNA to the ribosome P side after recognizing a start codon on mRNA bound to small ribosomal subunit. The large subunit then joins the small subunit and a second tRNA is recruited. As the mRNA moves related to the ribosome, successive tRNAs move through the ribosome and polypeptide chain is formed. So in this way, elongation proceeds with charged tRNAs sequentially entering and leaving the ribosome as each new amino acid is added to the polypeptide chain. Now this movement of tRNA from A to P to E side is induced by conformational changes that advance the ribosome by 3 bases in 3' direction. So the energy for each step, such as binding of a new amino acid tRNA to the A side and for its translocation to the P side after formation of the peptide bond along the ribosome, is donated by the elongation factors that hydrolyze GTP. So peptide bonds form between the amino group of the amino acid attached to the A side tRNA and the carboxyl group of the amino acid attached to the P side tRNA by an enzyme peptidal transferase integrated into the large subunit. So step one is after the amino acids needed to make the protein are properly bonded to each other and release factor binds with the complex when a stop codon enters the A site. So release factor then disconnects the polypeptide from the tRNA in the P site. The remaining components of mRNA and ribosomal subunit in the protein release and the process of translation ends. So the overview translation is that tRNA with first amino acid and the initiation transitional complex forms, and tRNA brings first amino acid in polypeptide chain to bind to start codon on mRNA. Then tRNAs bring amino acids one by one to add to the polypeptide chain, and release factor ignites stop codon, translational complex dissociates, and complete polypeptide is released, and the components are recycled. So that is the overview of translation. So in prokaryotes, met tRNA is modified by the addition of a formyl group, and it is F-met tRNA in prokaryotes. So do the same DNA sequences lead to the same proteins in all organisms? No. Due to differential mRNA splicing, identical DNA sequences may lead to non-identical proteins. There are about 150 known cases of deviations from the universal genetic code, with one or two codons being reassigned to different amino acids, typically in small organelle genomes, but occasionally in nuclear genomes as well. Okay. So coming to the conclusion of this question, the coupled translation happens in prokaryotes, and the answer will be increasing rate of protein synthesis. So if you have any doubts in this question, you can ask me. Okay, guys, if you don't have any questions, we can end the session today here, and we'll meet again next Monday, same time, 7 to 9. Okay, we can end the session today here. Thank you. Thank you so much. Thank you so much. We'll meet again next Monday, same time, 7 to 9. Thank you.